All right, folks, we got one real quick unit here over DNA technology, and this is one of the most interesting things about DNA, is that just slightly more than 60 years ago, we discovered the structure of DNA, and now we're altering it in ways that would blow your mind. We can do so many amazing things with DNA now. We can compare it to other DNA samples. We can find out who the father is in a paternity test. We can make genetically modified organisms to benefit society. It is amazing to consider all of the different things that we can do with DNA. So let's look at some of those technologies and see how they can benefit society. Today you're going to be able to explain how humans have altered DNA and used it to better society, and examine different techniques used to change or use DNA for the advancement of human technology. We can do so many amazing things with DNA, and it's important that I share some of those with you today. So, let's talk a little bit about the concept of genetic engineering. That is the technology that we can use to manipulate the DNA of an organism, and we can do several different things with this. We can just simply alter the DNA inside of an organism. We can insert or remove genes that we want to place inside of an organism, or we can make copies of genes. One of the most practical applications of this technology is making insulin. The insulin gene is actually found inside of a pig. They found this gene, cut it out or spliced it, and inserted it into a bacterial plasmid. That bacteria then produces the insulin. And so they have hundreds of thousands of bacteria making insulin now. So this is really important for medical technology. The genetic engineering to make mass quantities of insulin is incredible. And we can do other things with genetic engineering as well. So how do we use this? And here are some tools that we use. One is known as the genome, which is the total DNA in the nucleus of the cell, and that it's important to know the genome. A few years ago, the Human Genome Project, or being able to sequence the entire set of DNA inside of a human cell, was one of the biggest topics in science. We're then able to utilize this information and figure out what sequences code for what genes. We can then alter the DNA as we see fit. In order to alter the DNA, we have to actually cut the DNA, and that is done using what's called a restriction enzyme. This can bind to DNA and cut it at specific sequences. So you see here, the DNA or the restriction enzyme, EcoRI, is being used to cut the DNA sequence here. This can create what is called sticky ends, which have DNA segments without complementary pairs next to them, or blunt ends, which would just be like cutting the DNA right down the middle. So restriction enzymes are used to cut the DNA. We can then use whatever DNA we want and insert it into that plasmid near those sticky ends. It just provides an opening for us to insert whatever DNA we want inside of the cell. Some other tools that we can use, another one is known as gel electrophoresis, and this sorts DNA fragments based upon the charge and the size. What this does is this creates a banding pattern on the gel, and we can compare different samples of DNA. So we take some DNA and make lots of copies of it and insert a restriction enzyme that cuts it into smaller pieces. We then insert the gel, or insert the DNA inside of the gel, and then run an electrical current through it. Because DNA is negatively charged, it's actually going to run from the negative to the positive. So as a result, the DNA is going to move through the gel. The larger pieces of DNA will get stuck in the gel closer to the top, and the smaller pieces of DNA will move through faster. If they're the same DNA, the restriction enzyme will cut at the same locations, and you'll have similar bands that are present on the gel. Alright, so it's a way we can compare DNA samples in either paternity cases or crime scene investigations. Recombinant DNA is simply DNA that is found from multiple sources. So what we can do is take a bacterial plasmid, cleave the plasmid DNA with the restriction enzyme, so cut it into cut the DNA, and insert the new DNA inside of the bacterial plasmid. thus creating DNA that has the new gene inside of it. Now, eukaryotic cells, this is a little bit more complicated because they don't have plasmids, rather, they have chromosomes. So it's a little bit different and a little bit more complicated. So most of the experimentation is done with bacteria. But we've been able to make bacteria that produce insulin and even bacteria that glow in the dark. It's a really cool and useful technology that will benefit society for many years to come. Another is what's called PCR, or polymerase chain reaction. And this makes millions of copies of a specific region of DNA. So you hear a lot about crime scene investigations where there's just like a hair or a skin sample. How do they extract so much DNA from that? Well, they perform a PCR reaction where it actually makes millions of copies of DNA, and we can use that to analyze and see if DNA segments are similar or different. It just allows for greater research opportunities using small samples of DNA and making them larger. It's also used for forensic scientists and crime scene investigations as well. Things that we can also do is create genetically engineered organisms, and these are called transgenic organisms. And we can produce plants or animals with new traits that are resistant to disease, and some that can even for cosmetic purposes like glowing in the dark, and potentially even design our children. We could, in the future, be able to determine the traits of our offspring. So something to think about is the ethical implications. Is it right for us to be able to determine what traits our offspring have? Whether it's something like intelligence or something like eye color. Is it right to be able to alter the DNA of organisms for our own personal benefit? These are just some things that I want you to think of as we go through this, because I'm certainly not saying that all of these technologies are wonderful, amazing, life-saving technologies. Some of these have some very serious implications that need to be thought out before we fully implement these technologies. So think about those as we move forward throughout this unit. So hopefully today you have an understanding of how humans have altered DNA and used it to better society, and you're able to examine different techniques used to change and use DNA for the advancement of human technology. Again, DNA technology is a wonderful thing, but we need to think about the implications of using it, and whether it can be both good and bad is really important for society to discuss. So, hopefully you understand this a little bit better. Make sure you take your video quiz at the end of this, and we'll talk to you guys later. Have a great day, everybody.